I am Dr. Gamou Fall, a virologist working at the virology department at Institut Pasteur de Dakar. After my PhD graduation in France in 2009, I joined Institut Pasteur de Dakar first as a postdoc until 2013. Then I got a permanent position, and right now I'm a research associate with public health, training and research activities. The virology department hosts a WHO collaborating center for arboviruses and hemorrhagic fever viruses, as well as a yellow fever regional reference laboratory in the context of the WHO Yellow Fever Laboratories Network. And since last year, I am the lead of this yellow fever laboratory. I am therefore involved in all activities regarding disease surveillance, particularly yellow fever, as well as virus identification from mosquito pools as part of the entomological surveillance. Regarding these public health activities, I'm conducting since last year a surveillance project funded by NIH as part of a consortium with the Institut Pasteur International Network on two major arboviruses, Rift Valley fever virus and Crimean Congo hemorrhagic fever virus. At the virology department, I'm also involved in training activities by supervising master and PhD students from Senegalese universities, as well as organizing training workshops mainly for WHO and West African Health Organization. Regarding research, my activities are mainly focused on the analysis of the interactions between arboviruses and their mosquito vectors. And I'm using as models viruses such as West Nile, USO2 and Rift Valley fever. Indeed, my OWSD project is focused on the identification of mosquito saliva proteins that are involved in Rift Valley fever virus infectivity in vertebrate host. The problem we are facing here is that arbovirus disease have increasing incidence in the world, and control means are absent or ineffective for many of them. As the recent COVID-19 pandemic highlighted the vulnerability of health systems and the need to fill gaps on basic knowledge and control tools regarding emerging pathogens, it is really crucial to develop control means for arboviruses. Indeed, these arboviruses are transmitted by arthropods to vertebrate hosts through inoculation of virus-infected saliva into the skin of the vertebrate host. Interestingly, it has been shown that this saliva can enhance pathogen infectivity in vertebrate host. And in this host project, I aim to identify the mosquito saliva components that are involved in the saliva-mediated infectivity enhancement. So my motivation is that this study might constitute the basis to new control means based on the arthropod vector. Indeed, their characterization could lead to the development of therapeutics or transmission blocking vaccines to control the transmission of Rift Valley fever virus by the vector and its dissemination in vertebrates. This approach is very innovative, as traditional vaccines are usually based on the pathogen components